share some of the exciting new developments, albeit quite preliminary, that we have been able to produce from the Institute. And in this brief and necessarily broad presentation, I really hope to inject a scientific stimulus package, which is much needed, into this field of chronic fatigue syndrome. So to begin, there's a sort of graphic representation of XMRV on the left ahead of you. And on the right is an actual EM from one of the patients, demonstrating the presence of this retrovirus. Reviewing briefly the retroviral life cycle, you all recall that retroviruses are latent and they can be also active, and the techniques for evaluating latency and active infection are different and available also for this particular retrovirus. That, of course, is a HIV model that's presented there. With XMRV, it was important to differentiate it from all the other mouse retroviruses that are in the family of the gamma retroviruses. And this phylogenetic tree that was developed by gene sequencing demonstrated that this particular XMRV that we isolated from the chronic fatigue patients was similar to, but not identical to, the XMRV that has been demonstrated in patients from prostate cancer, which are represented by the VP62 and the VP25. You'll also note that phylogenetically, this particular group of XMRV is quite disparate from the mouse retroviruses. What this means somewhat simplistically is that there's been a genetic deviation from the other mouse retroviruses, making it extremely unlikely that this represents mouse contamination in the laboratory. Secondly, you will see that there's genetic differences in the strains that have been sequenced, which is what one would expect from retroviruses that arise in different locations and in different infections. This work was conducted by an unusual collaboration of the private sector, the Peterson Institute, the National Cancer Institute, and the Cleveland Clinic. And I bring that up to show what can be accomplished in a relatively short period of time with relatively limited resources, combining the expertise that no one of those entities would have had. The study cohort that was reported in Science came from the WPI National Repository. We've been collecting specimens for a number of years in -80 conditions. And the repository samples included specimens from Nevada, California, Oregon, Florida, North Carolina, and New York, as well as some international patients. The repository criteria were very simple: a CFS diagnosis. The age range was 18 to 75 years, and the patients broke down to 67% female, which represents the general gender incidence of chronic fatigue syndrome, with a mean age of 55. That's slightly older than some other reported series, and that's probably because they were drawn from practices such as myself, where we followed people for a very long period of time. There were 320 control samples that were roughly matched by age and sex and geographic distribution. I point out that these control specimens were not contact controls, laboratory workers, family members, etc., which would strongly probably bias the findings on controls, potentially. So many questions, of course, arose when we had this discovery. What cells are infected? What's the tissue type? Is the blood even the place to look? Should we look in bone marrow, brain, spinal fluid, etc.? The immediate question that arose in my mind is, how is it transmitted? And that's obviously a major concern for issues such as the blood supply. Then the critical question is, do infected individuals in fact mount an immune response? And if they do, can we measure that? And ultimately, can we manipulate that to the patient's benefit? Of course, the obvious question, are there interactions between this particular retrovirus and the immune system? Again, asking the same questions that were asked in HIV many years ago. Another question that, of course, is pertinent is, since the mouse retroviruses are very well known to cause lymphomas, leukemias, etc., may the human version of this, XMRV, be associated with the development of cancer as well? And then finally, given the findings, does this give us some thoughts and directions about more specific therapy than we've ever been able to come up with in chronic fatigue syndrome in the past? So again, the CDC criteria are well known to everybody, and I point out that even as early as 88 and 94 with the revision, there was an emphasis in this case definition for flu-like illness, for symptoms that suggest the possibility of a viral onset and perpetuation of the syndrome. Of course, we've been troubled by the fact that it's obviously heterogeneous, highly dependent on case definition, highly dependent on the observer in order to define the cohort that's being studied. And it's been said many, many times that that has to be clarified in all studies that are done. And the concept of clinical networking, I think, is absolutely critical to proceed, particularly if we have an agent like this that could be studied across regional borders. What intrigued me for many, many years was the fact that if you study the literature on patients severely affected with chronic fatigue syndrome, certain things stick out. For example, the RNase L dysfunction in the antiviral pathway has been reported by researchers from all around the world. The same finding with low natural killer cell numbers and function. Very reliable researchers such as Dr. Kleiman's have reported this over and over. And there's abnormalities of the innate immune system with activated T cells and production of inflammatory cytokines. And most everybody who has looked at this has reproduced those particular findings. So the question was, how does this new finding of XMRV impact what we've already known? Well, this particular gamma retrovirus is a small, simple retrovirus. It only codes for structural proteins, and hence it might present different problems with respect to therapy, for example. It might be much more difficult, and the existing HIV drugs may or may not be effective. Again, we know that retroviruses are not, in fact, ubiquitous, and they're not benign. And this group of retroviruses particularly are known to not be benign. And I would submit that HIV and HTLV1 are also not benign in the human population. I've just selected a few representative pieces of the science so that you can get a grasp of what we're doing and what this looks like. On the left, you will see 11 representative patients that were assayed for the GAG and the OMV, and you'll see that 68 of 101 that were assayed were positive, and positive with a very clean, distinct pattern. On the right-hand side, you will see a group of 11 controls where one control was positive utilizing the same technique. When you extrapolate that to the 320, you'll find that there were 12 positive controls, or a percentage of about 3.75%. And as a practicing physician, that's a grave concern to me, because that means approximately, if that holds up with other control groups, that approximately 4% of the population has evidence of this virus that we only knew about by running controls. So it opens a whole nother area of concern with respect to development of the disease, associated diseases, or the safety of the blood supply, for example. The question arose, in which cells could we find this particular XMRV? And this is a little bit complex, but basically we took stimulated B cells and T cells, and we're able to demonstrate the presence of the virus both in B cells and T cells. This represents just one patient, but was replicated in the other patients as well.